The overall goal of this fluorescent time-lapse microscopy protocol is to provide a method for studying biological cell processes that underpin the development and cellular differentiation in the sporulating filamentous bacterium Streptomyces venezuelae.
The described method provides an excellent platform to study cell biological processes that are central to the Streptomyces life cycle, including dynamic protein localization, polarized growth, and sporulation septation. The main advantage of this technique is that Streptomyces venezuelae sporulates in liquid, which allows us to grow the cells in a microfluidic device and microscopically monitor the complete life cycle.
This method demonstrates the immense potential of Streptomyces venezuelae as a new developmental system for the genus because it allows for live cell imaging of the differentiation of a multicellular mycelium into chains of unigenomic spores.
To begin, inoculate 30 milliliters of supplemented growth medium with 10 microliters of spores from the S. venezuelae strain to be imaged. For consistent growth and sporulation of the cells, use a baffled flask or a flask that contains a spring to allow for sufficient aeration. Culture the cells for 35 to 40 hours at 30 degrees Celsius and 250 RPM. When ready, you should be able to see the mycelial fragments and spores via liquid-mounted phase contrast microscopy. Centrifuge 1 milliliter of the culture in a tabletop centrifuge at 400 times G for 1 minute to pellet the mycelium and the larger cell fragments.